Thank you very much, Fanas, and thank you everyone for coming to this SASSB webinar. I note that we're starting 30 minutes earlier than normal, so we might get a rush of people coming in at 4 p.m. It's an absolute pleasure to have our speakers for today, and especially on the theme which continues our wish as the SASSB Council to have a diversity of talks which represent the diversity of fields that our membership cover. And starting last month with looking at dinosaurs and paleobiology, today going to bacteria. So not just extremes in size, but also perhaps time scales. But overall, I would say thank you very much and welcome everyone, and especially a big thank you to Fanas and the team at UP for hosting and coordinating this talk today. We hope that we'll be enlightened, we'll learn and grow. And over to you, Fanas, to introduce the speakers, please.
Okay, thank you. I also want to just welcome some members of ISME that's associated with South Africa and maybe some places in Africa. So the International Society for Microbial Ecology is having their large conference this year in Switzerland. And in preparation of that, we are also starting some talks and things in South Africa just to create some interest. And I'm the ambassador for ISME in South Africa, and with Tulani, we decided this would be an ideal opportunity also to link up with the SASSB. So thank you also to the bacteriologists and microbiologists with an ISME interest that who have joined us today. So before taking up too much time, I want to introduce our first speaker, and this is Luis-Miguel Rodriguez. I know him as Miguel, and I'm very happy that I could convince him to give us a talk, because I've worked with him and have been in meetings with him, and he's always impressed me very much. Especially his enthusiasm for things and his willingness to help people. And also his knowledge. You will see also from what he's doing today that, he's got it's one of those biologists with a very good computer background. And that's not always so easy to find, but it's wonderful to have you. So Miguel started his training in Colombia, but he's a person that likes to travel the world. So from Colombia, he then studied in France. He did an MSc there in applied informatics, and then he did his PhD at Georgia Tech in the States. He had a post there for a while after that, and now he's moved to Innsbruck. So he was telling me he has to learn German now in order to have a conversation with his colleagues, and to be able to order beer probably. So, but you will also see from his interest that, as I've said, he's a biologist, but also bioinformatician. I work with him in two contexts. One is on Salinibacter, where we have an international group and where he's made some nice inputs, and the talk by Catherine is coming from that association. And then the other thing where he's put a lot of energy in is what we call the SecCode initiative. This is an initiative by the bacteriologists and Archaea people to create a taxonomic code that will be based on sequence data. And Miguel is the big driver for the registry. So he's really created the registry where the names will be registered, and that will be the official place to register these names, and they will then have standing in nomenclature, something that we've asked for years but have not been able to achieve. So there he's also been doing a lot of work, but he's going to tell us today about some of the work that he did at Georgia Tech, and many of the bacteriologists might know about the MiGA or the Microbial Genome Atlas. So over to you, Miguel.
Thank you, Fan. Thank you for that very kind introduction, and also thank you for the invitation. I'm very happy to be here. Okay, so this should be full screen, right? Perfect. So good afternoon, everybody. So as Van mentioned, I will be talking today about indexing the genomic diversity of archaea and bacteria with MiGA, or the Microbial Genomes Atlas. You can just a matter of Fanus did a very kind introduction, but just if you want to be in contact with me at any point, this is here my web address and also my Twitter handle. And I'm an assistant professor at the University of Innsbruck at both the Department of Microbiology and the Digital Science Center. So before I get started, I wanted to just briefly mention that any screenshots or even if you want a copy of the slides, that's all allowed. All this slideshow will be it's available under the Creative Commons attribute or by 4.0 license. So you're free to take snapshots if you want. And before I get to tell you what I'm doing, I wanted to tell you with whom I'm doing this. Usually these acknowledgements stay at the very end of the presentation, but I don't enjoy doing this in a rush. I just want you to know that all of the work I will be presenting here has been the result of many collaborations with the Constantinidis lab at the Georgia Institute of Technology and some former members of the laboratory, Dr. Zemenzi, Dr. Luo, Dr. Orellana, and Dr. Karthikeyan. Also, there have been many of these results are collaboration also from other collaborators at Georgia Tech, some collaborators at Michigan State University, and some collaborators from around the world. Of course, as usual, I would like to thank, of course, the funding agencies that made this work possible. So now I'm gonna be talking about classification, right? And I know this crowd is very familiar with the concepts of classification, but in the specific case of prokaryotes, of bacteria and archaea, we kind of have to go back all the way to the beginning because the first assumption that we make when we're classifying anything, and specifically when we're classifying organisms, biological entities, is that there is some sort of unit to classify, right? Some sort of natural unit to classify, which in animals and plants has been for the longest time the dominant view has been to use the biological species concept, right? Which is the idea that organisms that are sexually isolated and that interact sexually between them, they form a unit, right? And that unit we call species. We hit a wall, however, when we analyze or when we study organisms that reproduce asexually, like is the case of archaea and bacteria. However, there is this one glimmer of hope, this observation that has been coming again and again since really since the early 2000s. It has been observed by many researchers, chiefly and very early on by Dr. Constantinidis, working with both the laboratories of Ed DeLong and also the laboratory of Jim Tidji. In this observation is that it would appear that there is discreteness, a sequence discreteness in the populations of bacteria. So when we take a genome, here represented in, this is a piece of a genome, and the x-axis here in this top graph represents the position along that piece of the genome, and then the y-axis represents identity of different sequencing reads, right? The identities to that piece of the genome. And one thing that we have seen and again in different environments is that it would appear that the identities are either very low, meaning like somewhere below 85%, or very high, somewhere above 95% of identity. But there is a gap. There is some discontinuity. So this discontinuity, that doesn't mean there is nothing in between. It just means that the density is so much lower that it would appear that this corresponds to actually some sequence discrete or some discrete populations that actually form naturally out there in the environment, right? Another way of representing this is by looking at a histogram of coverages, and in a histogram of coverages we usually see, again, either values that are well above 95% or well below 85%, but there is a genetic discontinuity in between. And this is something that we see for small pieces of genomes or for entire genomes in very different populations. The example at the top is an example from marine microorganisms. The one at the bottom is an example from freshwater microorganisms. And we have been able to reproduce this genetic discontinuity or observe this genetic discontinuity in many other organisms. Now, interestingly, there is this algorithm that was originally developed by Kostas Konstantinidis called the average nucleotide identity, that is basically a technique to measure the identity between any two genes, right? When we are classifying things, of course, the first thing that we need is units. And I hope I convinced you to some extent that there seem to be some units, some natural units out there in the environment. But the second thing we need is we need to be able to compare between them, right? And one such comparison is the average nucleotide identity, which is basically a methodology of taking an entire genome, breaking it into little pieces, taking the second genome, breaking it into little pieces, and then comparing those pieces and extracting or calculating the average nucleotide identity between all reciprocal base matches in those two genomes. And interestingly, when we do that for large scale collections, for entire databases of genomes, thousands or tens of thousands of genomes that are now available for Archaea and bacteria, what we see again is this discontinuity. We see again that most pairs of genomes have an identity that is well above 95% or well below 85%, with a big gap in between. And not only that, but actually these high identity pairs mostly correspond to comparisons within the same species, meaning species as in the way that we have been traditionally named species in bacteria and Archaea, which is it's a combination of phylogenies and phenotypic characterization. So it seems to correspond very well to within species, like those values that are well above 95%, and the values that are well below 85% seem to correspond to comparisons between pairs that are between species, that are from different species. So now we have both a conceptual framework in which to frame our classification techniques, as well as an actual tool, an algorithmic tool that allows us to actually compare genomes, entire genomes within that framework. Of course, like any important scientific observation, this is not without controversy. There has been some controversy around whether or not this ANI gap that I just showed you is real or not. We ourselves have been showing different ways of displaying these histograms by controlling for sequencing time, by controlling for technology, controlling for taxonomy, and we were able to recover this gap very clearly. But there is some evidence from simulation showing that, in fact, a similar discontinuity could be reached by a bias in isolation, by an isolation bias. We believe that the evidence from metagenomics that has no isolation bias is a fairly strong evidence of the existence of this discontinuity. But if you're interested in this this fascinating debate, I invite you to take a look at all of these publications that are, you know, comments on comments and replies. Now, this whole framework that I have been telling you about to compare between genomes sounds great, but do we even need all this fancy equipment? Do we even need to compare entire genomes? Probably those of you that are familiar with prokaryotic taxonomy know that we actually have a really good tool already, that is the ribosomal RNA gene, the 16S, right? The 16S ribosomal RNA gene is already a really good, very robust gene that allows us to build robust phylogenies. It allows us also to build an entire scaffold for the taxonomy of prokaryotes. So why do we even need to go into entire genomes if a single gene is already doing a good job? And the answer is twofold. On one hand, and this is what I'm showing here, on one hand, 16S works great when you have more distant organisms, when you are working at the family or order or beyond levels, or to distinguish domains, which is actually the original claim to fame of 16S rRNA, and it is that Carl Woese originally proposed the 3-domain model using ribosomal, the small subunit of the ribosomal RNA. But when we go all the way down to species and to within species, the identities saturate. And we have certain problems. We have, for example, different copies of the 16S within the same genome that might have very low identities. So we could inflate the diversity if we consider those different species, even though it's the exact same genome, the exact same cell. But the opposite is also true. We can actually underestimate diversity because there is a saturation level near the species level. So very many pairs of distinct species actually have almost identical or almost identical 16S sequences. And this is a study that we did trying to identify by how much do we underestimate the diversity for different taxa. And the answer is what we were hoping to get out of this analysis was a single value, right? Say we are underestimating by 75%, period. Easy, we can fix this by simply multiplying the measures of richness by that. What we actually observed is that if we compare genome species to 16S OTUs, depending on the taxonomy of the organisms we compare, we can get very different ratios, very variable ratios. Some of them underestimate, some of them overestimate, some of them underestimate. And so that's very concerning, because then we cannot really resolve the species level objectively at the 16S level in a way that is easily reproducible and automatable. that's why we are using average nucleotide identity instead. Now, the average nucleotide identity, well, and by the way, sorry, I said there were two reasons. The second reason is that we are now at a point at which we are able to recover almost complete genomes out of metagenomes, and that has allowed us to study the uncultivated majority in great detail. But these kind of genomes that we extract from metagenomes, called metagenome assembled genomes or MAGs, they often don't have 16S because the very highly conserved regions are very hard to assemble out of a metagenome. So that's the second problem. In some cases, in many cases, we don't even have a 16S. Now, the ANI is great because it allows us then to have an objective definition of the units that we want to call species. And not only that, we also we can also do it with great resolution, all the way down to very closely related strains of the same species. But it has a problem, and is that it drops very quickly. It's based on a fraction of the genome that it drops very, very quickly. So by the time we are comparing between families or so, we no longer have enough shared genome to actually calculate ANI. So instead, what we do is we go back to the genome. Instead of shredding the genome into little pieces, what we do is we calculate the genes and predict the proteins out of that, and then compare the proteins directly. And of course, the conservation at the amino acid level is a lot higher. And this alternative algorithm is called the average amino acid identity, or AAI, and that allows us to actually compute distances at any within the entire range of the genetic diversity within prokaryotes. Now, all of this is great. All of these tools individually are great, but they have a giant problem, and it's that they don't by themselves scale very well, right? Not because they have a fundamental flaw, but simply because they require a lot of manual intervention to just calculate these distances and actually compare genomes. And it takes some scripting, and it takes some bioinformatic knowledge. And that's a problem. That's a problem because we want this framework to be used by biologists. So what we decided was, well, if there is no framework that allows us to do this at scale, how about we work on it? And that's exactly what the Microbial Genomes Atlas, or MiGA, is. So MiGA takes as input any genome or even a metagenome, processes that data, does the assembly, everything, the gene prediction, everything that you may need, and importantly, it evaluates the quality of the data, which is a very important part of the quality check is a very important part of this process. Then for those genomes, it can calculate whole genome comparisons at the highest possible resolution. So it will start by doing by using only conserved genes. If that's the distance is large enough, then that's good enough. Then if not, then it will go into doing a full genome AAI. And then if that is close enough, it will even calculate the entire average nucleotide identity to distinguish between species and subspecies. It implements a novel search algorithm to allow for the efficient search against thousands or tens of thousands of genomes in a reasonable amount of time. So we can build databases that we index, and then they are queryable by your own genomes in a couple of minutes, as opposed to the hours and hours that it will take to actually compare against all of those 10,000 genomes. And then it has a robust empirical distribution of identities related to taxonomy to provide both taxonomic novelty tests, so a p-value associated, an empirical p-value associated to how novel your genome is at every rank, and also an accurate taxonomic classification. It will also have a p-value associated at every rank for the classification of your genome. This is a big overview of what of how the entire pipeline within MiGA looks like. And I know this is scary, but the only thing that I want to show here from this entire chart is that the inputs can be either genomes or metagenomes, and you can start querying it all the way up from the raw reads of the genomes or the metagenomes, or from already assembled genomes or assembled fragments of metagenomes. And then it will take over from there. Okay, now this was our initial implementation of MiGA that worked fantastic. We had this engine, and then we started getting requests on how to use it in different environments. And this kind of organically grew into an entire portfolio of products that we have now built on top of MiGA. So of course, the first part of it is the actual code base, the part that the engine of MiGA, the part that works, that does the heavy lifting. The focus here is efficiency and flexibility. We want just a very basic engine with a thin command line interface that just allows to do anything either from the command line or directly through a Ruby API. And this has gone through many different iterations. We use semantic versioning to make sure that we always are backward compatible, and we are currently in version 1.2. So this is a very robust project already. Just to give you an idea of how large this project is, this is a sequence code analysis where I just want to show you that the entire, or the basis, the bare bones of the MiGA project is actually estimated to be a project that would cost commercially $1.1 million to produce, and it will take over 14 months of dedicated work by 7 people working on this, as full time. So this is the scope of the engine. Now, our first product on top of MiGA code base was MiGA Online. So MiGA Online allows for, the focus here is the manual inspection. So it's a web interface that allows us to actually explore the results that MiGA provides. This here is our this can be freely accessible at microbial-genomes.org. You do need to create a user, but that's it. Everything is freely accessible. And just so you know, this is a product that has been going through iterations for several years now. This here on the left is the new monthly genomic query. So we typically receive somewhere in the order of 1,000 queries per month. And this here are some of the hallmarks of our development. When we first announced MiGA publicly, when we published the first manuscript in nucleic acid research, and when we published the most recent manuscript in the Burgess manual. And we have well over 1,500 users now currently active, registered in the system. And this curve keeps growing, as you can see. Now, what can you see in MiGA? What can you get from MiGA? So the first thing is, of course, I have been talking to you about classification. So that's, of course, the first thing that I will show you. So when you upload your genome, and as I mentioned, this is freely available. You can just upload the genome and hit run, and it will do everything for you. When you upload your genome, the first thing that it tells you is what are the closest relatives in the database. It will mark the ones that are type species with a capital T after the name, and then it will tell you what's the average amino acid or the average nucleotide identity to them, and it will also show the taxonomic classification. So of course, just because the best hit is in Mirania, it doesn't mean that your genome is in Mirania, right? So it will calculate a p-value associated with the classification at every single rank, and it will calculate also a measure of novelty, how much. If you wanted to describe this organism, this genome, as a new species, for example, you could have actually a statistical support on how likely it is that this is actually a new species, right? Or a new genus or family, etc. You can also see different metrics of quality. I don't really have the time to go into many details, but it will just to show you that we try to make it as user-friendly as possible, even trying to get an indication of what is a very low degree of contamination, or an intermediate or a high degree of contamination, summaries of quality, references with respect to RefSec for gene length and genome length and these things. And then we provide additional, more advanced features, like, for example, this here, there is a window-based taxonomic classification that will allow us to very quickly check manually our genomes to identify things like clear cases of contamination that can be moderate or high contamination. As you can see here, this zebra pattern, that's one of the telltales of contamination, but also when we classify a big chunk as something completely different. And by the way, this thing here, this example here is an example out of the NCBI, directly out of the NCBI, that is called, that in NCBI is called a Streptococcus pneumoniae. But as you can see, the classification based on any part of the genome should be a Bacillus cereus. It just happens to have a small contig of contamination that includes 16S, and apparently this was used, this was classified on the basis of 16S alone. Now, these are I'm going to actually skip these, but these are some of the things that you can do in the MiGA online interface. Now, this whole interface is available. We provide the computing power behind it, and you can just upload your things, your genomes here. But we know that not everybody trusts public servers to handle their data. So we actually made the entire interface available under the artistic license 2.0. So this is open source, and you can just install MiGA in your own servers and install MiGA Web and have your own interface. You can have an entire clone of MiGA Online in your own servers, in your institutes, or even in your laptops. The problem, of course, is that it requires some configuration and some installation, and that's not super easy for everybody. So what we're trying to do now, in collaboration with Michigan State University, is to produce containers supporting both Singularity and Docker, such that you can, by simply installing Docker in your laptop or Singularity in your high performance computing center, you can basically have MiGA with the stroke of a couple of commands, or even with a couple clicks. So this is something that is coming up soon. This should be, we are already in beta testing. If anybody is interested in testing this in your own institutes, please do contact me, because we will be happy to include you in our beta testing group. And this is, of course, tied to, once we have containers, we can also push this all the way to cloud computing, right? So we're also working together with Michigan State University again to put this as a cloud resource, so you can also use it, for example, in the classroom, right? So you don't have to have fancy high performance computing capabilities, just a relatively small budget for cloud computing, and then boom, you have MiGA in your classroom. And finally, I mentioned that MiGA Online the focus is very detailed, careful examination of one or few genomes. But what if you have not one or few genomes, but 100 or a few hundred genomes, which is a problem that it's a situation that we hit very often these days. Well, if that's your case, we also provide a web-based interface called MiGA at Exceed, or the Exceed Gateway, that is available at exceed.microbial-genomes.org, where you can actually upload a bunch of genomes, as many as you want. We don't really have any limit here. You could upload as many as you have. Of course, the more you upload, the more you have to wait for the results. But you upload them, and then you can directly download the entire results. Not just in MiGA Online, you can browse them, in live. You can browse all your results online. In MiGA Exceed, you actually get to get all the results down and do whatever you want with them. And this is supported by Exceed, which is a program of the National Science Foundation of the United States, and it runs in the supercomputer, in a supercomputer called Expanse in the San Diego supercomputer. So some of the things that you can do in MiGA Exceed is build your own databases, do genome typing against against large databases of intra-species collections, genome classification against general databases, or genome dereplication, which is a very common task these days, mostly from metagenome assembled genomes. Now, we have a big problem I have been talking about databases, but we have a big problem, and is that biased databases produce biased results. So you probably already know who this gentleman here is. There is this one experiment in which an algorithm was developed to try to enhance a blurry picture of a person by learning how people look in general. So you probably already know who the gentleman at the top is, and here there are some examples. When some researchers, some external researchers, fed these images to the algorithm, they obtained these results. Clearly, there is a very obvious bias, and it's that the algorithm was learning how faces of white people look. It wasn't learning how faces of humans look, it was just learning how faces of white people look. And so the reconstructions of all of these blurry images are reconstructions of white male and females. So we can actually fall into the same problem. If all our databases are built on the basis of just proteobacteria, everything that we will see will be probably biased towards proteobacteria. So we need more complete databases. So this is something that we have been working on. I don't really have the time to go into all these studies, but we have been working on the identification, classification, description of microorganisms using techniques independent of cultivation. We have described taxonomically some of these species. This is the work of Dr. Zemensi at Emory University. This is here another description, Macondymonas diazotrophica, the work of Dr. Karthikeyan at San Diego's, California State, the University of California in San Diego, sorry, which is actually a very interesting example in which we were able to identify a MAG out of metagenomes, and then using the metabolic reconstruction of that genome, we were able to isolate it later. And interestingly, this microorganism was actually something that we had been expecting to find in oil-associated environments, but we had never seen, which is an organism that can both degrade alkanes and fix nitrogen. It had been theorized that such organism should exist, but we had never seen it. And once we found this organism, we actually found it everywhere around the world. So it was hiding in plain sight. We just didn't have it because we didn't have it in the databases. And this is, MiGA has been used in the past years to describe new taxa at every single level, from phylum all the way down to species, many of which are actually using culture-independent techniques. So not all of these, these are just some papers, some recent papers that have been using MiGA to describe new taxa. And then just to close, I would like to tell you that, as I said, we are interested in growing this database, reference database, by identifying high quality genomes and describing them. But of course, a big part of that description is proper nomenclature. So we are actually working, as Fanus introduced at the beginning, we are working on the possibility of describe and nomenclaturally describe new microorganisms using genomes without the need for isolates. And this is something that is called the SIG Code Initiative, which I invite you to take a look at the sigcode.de. We are trying to put together a sensible collection of rules and associated computational techniques to make it, to democratize the access to descriptions, nomenclatural and systematic descriptions of new taxa in prokaryotes independent of cultivation. So with this, I would like to thank you very much for your attention. I would like to thank our collaborators, of course, the institutions, Georgia Institute of Technology, the University of Innsbruck, and Michigan State University. A large chunk of the funding comes from National Science Foundation at the US. And here you can find, of course, more information in the website, microbial-genomes.org, and in the publication, in some recent publications where we describe some additional features associated to MiGA. Okay, so thank you very much.
Thank you, Miguel. That's really, well, as I say, as a bacteriologist, it's close to my heart. Very nice to hear and see. So it's now open for questions. So if you have any questions, just raise your hand, indicate that you would like to ask the question. You can also post it in the chat line, but it's easy to just unmute yourself and ask the question. I see that Nicola has got a question. Over to you, Nicola.
Hi, yes, thanks, Marnus, and thanks, Miguel, for a really interesting, enlightening talk. So I mean, I work on macroorganisms, so I mean, it's even more interesting and new to me. And I think, you know, obviously, I think this holy grail of sort of an objective way of delimiting species is something that all biologists are intrigued by and are looking for. And I think your on that MiGA, those p-values for fit into different taxonomic ranks, I think is going to be something that, well, I imagine already is something that bacterial taxonomists are extremely excited about and are making use of. And as far as I know, there's no equivalent system for macroorganisms. So I just wanted to find out a bit more. Obviously, that p-value is completely dependent on the assumptions of whatever algorithms you're using. I just wondered if you could talk a little bit more about that, whether it's purely based on identity or whether you use different kinds of investigations to get those inferences.
Yeah,
different kinds of analyses.
That's a very good question. So this is, so to answer shortly your last question, this is based exclusively on the distribution of average amino acid identity. So identities, genome-wide identities, as we observed in a reference well-curated collection. So for the different ranks, what we do is we calculate what's the distribution of AAIs, and then we calculate what number of organisms, what number of pairs, sorry, or comparisons are within the same taxon versus between taxa at every single rank. And then what we calculate really is the probability that any given pair of organisms is not in the same taxon at the given rank. So this probability is basically just based on what we observed in the databases for organisms that are that don't belong to the same taxon, but that have an AAI that is greater than or equal, or sorry, lesser than or equal to that AAI that is observed in the query, in the online query. So this is entirely based on historical classifications, of course, and so there is an argument to be made that, well, that's not a very natural way of defining it, but what we have observed is that there is a clear discontinuity at the more or less at the species level that I hope I convince you of, and then there is a relative continuum of distances above that. It seems like around the genus level, there might be something. It seems like, for example, at near the family level, there might be more functional conservation than expected by chance, but we're not using we're not using any of these definitions because there is basically no natural definition of a family or no natural definition of an order. What we want to do with MiGA is to reflect how it has been historically used, each one of these terms, and try to converge into a narrower, hopefully narrower and ever narrower definition for each one of those ranks, so the overlap is smaller. So our hope is that the more people use MiGA to classify their organisms, the less the overlap will be, but not because there is some natural gap or something, just because there is a historical convergence. Yeah.
Okay,
Thanks,
the next question is from Diego.
Thanks, Miguel, for that awesome talk. I actually have some question that someone just posted on the chat. I just wanted to know how does this compare to, say, for example, GTDB?
Yeah, that's a very good question. So, and this goes back to the question from Nicola, and it's the idea that GTDB is a database that has reconstructed an approximated maximum likelihood phylogeny on the basis of a few genes, a few marker genes, to serve as the scaffold for the classification of microorganisms, of prokaryotes specifically. And what they do is, what they noticed is that actually there seems to be, once you apply some transformations to those distances, which is something that they call the RED, there seems to be a certain regularity in this transformed distance space of every rank. So what they do is they use that to impose complete regularity, and they reclassified a bunch of organisms to fit these regular definitions. So I always like to think of classification problem as well. Classification is the language that we use in biology. So I like to think in terms of linguistics. And in linguistics, you have these two schools of thought. There is the descriptive linguistics and the prescriptive linguistics. So we are the descriptive linguistics. We are trying to see how people have historically used each one of these ranks. GTDB is the prescriptive linguistics. They're trying to tell people how it will be more uniform and how it will be more homogeneous to classify organisms. I don't think either of the two is necessarily superior to the other, but it's a very fundamental difference in the approaches.
Yes, thank you for that. Just as a heads up, I spoke to Donovan Parks, who is involved in the GTDB, and later, probably in September, he would give us a talk here at SASSB, because I thought that that approach of sort of normalizing evolutionary distances might be of interest to the people here as well. So just a heads up, something to look out for in the future. Any other questions? Let me just see in the chat. Lonnie said, well, he's answer, he had the same question as Diego. So anybody else that would like to still ask a question? Sorry, I missed Mutama. I missed that one.
Yes, thanks. So, I've typed the question on just on the societal attitudes and approaches on what constitutes enough information for describing species. I'm wondering whether the Microbial Society has embraced this new revolutionary approach, which to me is fantastic. You having still resistance like we had especially in study of plants, where use of DNA barcodes, even up to now, is not widely accepted.
Yes, so it has been accepted a lot faster than many other such ideas that redefine fundamental units. In general, up until maybe a couple decades ago, it was generally accepted that the gold standard for species distinction, for species distinction or classification between bacteria and archaea was the DNA rehybridization. Together with a battery of phenotypic analysis. It's, I believe, probably works from the group of Kostas Konstantinidis and collaborators are probably the only people that still cite these techniques as the gold standard. From what I've seen in the published literature, ANI has become the de facto gold standard for the distinction or classification at the species level. Not so much the AAI. There is still some debate on which one is what's more useful, if it's more useful to have a raw distance like AAI or to have an elaborate phylogenetic reconstruction, and there are very good arguments for both sides. And AAI has gone a long way and it has been generally accepted, but there is some debate there. But at the species level, ANI seems to be the de facto gold standard now in the community, yeah.
Yeah, just to come in there, I think our biggest battle now is that people use it as a strict cutoff. You know, if it's 95% below, it's 2 species, it's above, it's 1 species. And we often have to then say, well, the phylogeny is a bit more subtle. You know, it's around 95, but please don't use it as a cutoff. So we end up with subspecies instead of having, you know, proper species that have separated in their speciation. Nicola has got some extra questions. She says, is the genetic gap also visible in other organisms, macroorganisms? I don't know. The genomes is probably
so we are right now. The last version of MiGA implements a novel algorithm for the estimation of AAI. That is a lot faster. That's it, and it also resolves better distances near the root. And this new algorithm, which is not yet published but is already part of MiGA, is called Fast AAI. We have been exploring the possibilities of actually using expanding Fast AAI to both eukaryotes and viruses, and it looks like a very promising avenue. Up until now, we were just not able to calculate distances between eukaryotes in a reasonable amount of time, and we wouldn't be able to scale up to the hundreds of thousands or even millions of genomes that we would need to have any decent coverage of the viral diversity. So just up until now, we were limited by software and by hardware, of course. But with the development of Fast AAI, this is definitely something that is in the horizon for us now.
And her second question is, do you also have plans for future refinement, for example, refinement in response to detailed case studies or differential weighting of different genes or parts of the genome?
Oh, yeah. So we don't, but that is a statement that I need to qualify. So within Fast AAI uses a few marker genes to estimate the whole genome AAI, and we have identified some genes that are just more informative than others, and we are exploring the possibility of differential weighting to improve the estimation of AAI. But at the whole level AAI, we don't have plans for differential weighting for 2 reasons. One is that this is a meant to be a universal metric that can then be refined by more by for example proper phylogenetic analysis, not necessarily just approximated maximum likelihood, but actually proper reconciliation of gene trees. So this is the first reason, that if we were to do that, we wouldn't be able to deploy it at large scale across the tree of life. We know from recent work, for example, from the Borg group and from the Coelho group, that the large majority of groups of proteins, the large majority of protein families are actually unique to a species or maybe a genus. So we wouldn't be able to do that other than for a small handful of genes. But we can do it for the small handful of marker genes that we use to estimate AAI, and that's something that we're working on. Yes.
Thank you. Any other questions? So,
oh, by the way, sorry, I said I had two reasons. The second reason is that a small fragment of the genome actually already estimates very well the AAI. So there is, when we go all the way down to more or less 20% of the genome shared, that's when things start breaking down. But as long as our genomes are complete to more than 40%, we're good. We don't really need to distinguish between genes.
Okay, thank you. Any other questions? Then if there are no questions, again, thank you very much, Miguel. It was really a privilege to have you here. And as I say, one of the things that we realize in the Society for Systematic Biology is that different people working on different organisms always find something interesting, what others are doing, and something that you can apply. And that's the real benefit from having this association. So thank you very much. And there is a following article that Lonnie has sort of added, and highly impressed eukaryotic person as well.
Thank you.
Thank you very much for your time. We have started earlier this today because Miguel has got other commitments afterwards, and therefore he will disappear soon to be able to fulfill those commitments. So again, thank you very much, Miguel, and it was really enlightening and very nice to have you here with us and for you to share with us your ideas and how it's developed. So thank you very much.
Thank you, Faris, and thank you for the invitation.
It's a pleasure. Okay, so now we're going over to our two student talks, and the first student talk will be by Catherine Brink, and she's going to talk about the genetic structure and diversity of a Salinibacter ruber population. Now, Catherine last year completed her honors in bioinformatics at UP. She worked with me, and that's the reason why she's here today, because I really like the work that she was doing. It's quite interesting in terms of we're moving now from the overall diversity of bacteria to the diversity within a single species, within a single population. So I thought that would be quite interesting. And so this year she's in limbo because at the later this year in August, she's moving to the States, and she's going to join Kostas Konstantinidis group there to start a PhD. So she's going where Miguel is coming from. So there's a bit of a loop. So if you're ready to share, Catherine, over to you.
Thank you, Prof. Can you see the screen and hear me properly?
Yes, we can.
Okay, awesome. Good afternoon, everyone. Thank you very much for the introduction, Prof. I'm Catherine Brink. And as you've heard, in the last year or so, I've been fortunate enough to be part of a project studying the diversity in Salinibacter ruber populations. There are quite a few reasons why we might want to study this diversity. For me personally, a big motivation was the fact that microbial species don't necessarily have a clear definition comparable to the species definitions for macroorganisms. Not having a clear species definition makes it very difficult to properly identify these species for medical or industrial applications. Understanding this diversity will also allow us to see how selection, drift, and recombination affect populations and how these processes interact with each other in those populations. If we're lucky enough, we can even use this diversity to understand how the environment affects the structure and the functionality and the adaptations within populations. In order to understand diversity on a larger scale, it might help us to start at a very small scale. But unfortunately, most natural environments like soil typically harbor too much diversity to study population dynamics properly. Extreme environments like the solar sultans that we're studying have a much more limited interspecies diversity, which makes them ideal for studies on microdiversity and evolution. Two major regulatory factors in this environment are light and salinity, which are both easily manipulated by shade netting or dilution. Salinibacter ruber is the most abundant bacterial species within this environment. We collected samples from a solar sultan in Santa Pola, Spain, and then used this to extract a metagenome as well as culture isolates. The isolates were grown at 3 different salinities from 20 to 30% salt, and then we extracted genomes from those isolates. This left us with a data set of about 63 genomes that we supplemented with 2 reference strains from the NCBI database and an outgroup from a secondary Salinibacter ruber population in Mallorca, Spain. Overall, we had a data set with 66 isolate genomes and a metagenome to play with. Unfortunately, the annotation for these isolate genomes was pretty bad, with only about 11.6% of the total genes having a defined function. The core genes were slightly better, with just over half of them being annotated, so we focused on this for defining the start of our population diversity. We filtered the annotated genes for deletions and that's left us with about 618 genes that we could use to produce individual core genes that we then combined into a consensus gene tree, which you can see on the slide at the moment. What you can see here is that we can broadly define about 5 rough clusters in the population. We wanted to make sure that we identified the correct groups. So we ran a script that relates the number of clusters to the branch lengths and found that we obtained a local minimum at 6 clusters, which means that we were right. And I get to do my favorite thing ever, color coding. We use these colors to base the rest of our studies on, since we wanted to see if the population structure was consistent over a number of numeric and phylogenetic hypotheses. And I use the same clusters throughout the presentation for comparison purposes. The first alternate hypothesis that we tested was using FastTree instead of RAxML to generate the individual gene trees. We wanted to see if a different phylogenetic model would give us any different results in the population structure. For the most part, the clusters were actually very, very similar. And the only big difference was these 2 strains, which clustered with the orange group in our previous tree. We found that these specific genomes actually tend to jump around between the blue and orange group, depending on which clustering algorithm you're using, which may be an indicator of recent gene transfer between the groups. We next use SplitsTree4 to generate a phylogenetic network. A phylogenetic network is able to represent reticulate events like recombination, hybridization, and gene loss or deletion. We can see the same clusters again, and most of them have very deep roots in the network. The 2 strains from earlier cluster again with the orange group. One thing of note in this network is that the patterns within the clusters aren't as similar as you might expect. For example, this pink group is highly reticulated and very closely related, but then you look at the green group right next to it and it's much more regular. There's this variation between the clusters as well, which is interesting because it may indicate that there isn't a singular evolutionary process responsible for the diversity in the entire population. The overall ANI score in the population was 98%, but we wanted to see if we could numerically evaluate the population structure using the individual scores. In the heat map, darker blocks represent a higher ANI score, which means there's a higher similarity between those genomes. As you can see, we have these blocks in the heat map, and this shows you that there's groups of organisms or groups of genomes that have a higher similarity within them than between them. When we extend this clustering, we see that the blocks match up pretty well with the clades that we identified phylogenetically. The only differences really are this split in the blue group, and then this strain that clusters with the orange group and was an outgroup in the original tree. In this case, the two odd genomes that I was talking about earlier also cluster with the orange group. We were starting to get confident in ourselves, so we decided to expand our population and created a new data set that consisted of our 63 genomes from Santa Pola, an additional 30 genomes from Mallorca, 9 genomes from South Africa, and 6 genomes from NCBI. We ran these genomes through the UBCG pipeline, which is able to identify a set of universal core bacterial genes that we could use to generate this tree. The first thing that we noticed is that the groups were pretty intermixed. We're not seeing groups clustering based on where they were located. So we're not seeing a Mallorca group, and we're not seeing a South Africa group. They're all mixing together with the Santa Pola isolates. The next thing that we see is that we identify the same monophyletic clades again. And this can sort of imply that the structure isn't necessarily unique to the Santa Pola population, but might actually be a property of the species as a whole. All in all, these trees tell us that we were able to identify a distinct and reproducible genetic structure, even though these strains were more than 97% similar. The structure was maintained at the species level and could not be predicted based on geography and did not align with geography. And the clusters themselves vary and may not necessarily be governed by the same evolutionary process. We were in the unique position where we had access to both metagenomic and isolate data for the same population, and we took full advantage of it. We found that the average sequencing depth of all of the clusters was quite similar. They all have a depth of about 16 times when we look at breeds that are 95% similar, and they all have an ANIR of about 97.8 to 97.9%. The equal depth tells us that the metagenome was able to capture the full diversity of a population. We didn't lose any information about a specific cluster during the metagenome sampling. We can see this in the recruitment plots as well, which show very similar patterns for all of the clusters. This means that we were able to capture the full range of variation within the population through the metagenome. We could also use the data to check the abundance of the clusters in the metagenome. And we find that for the most part, the clusters are present in roughly the same amount in the metagenome and in the environment, except for the orange cluster, which is significantly more abundant than the others. Both of these conclusions are very interesting because, for one, it shows us that even though these clusters are distinct and very, very clear in the population, they still coexist. And then for another, this rule does not apply to the orange group. So what makes these strains so special, and why are they able to excel over the others in an environment that is as homogeneous as a solar solitone? That's unfortunately not a question I can answer just yet, but the opportunity to compare our isolates to the metagenome has allowed us to make important conclusions. First, we can see that the metagenome is able to capture the diversity of the population and represents each cluster equally. And secondly, we find that most of the clusters are able to exist, except for the one which occurs at a higher abundance. We know that this population is diverse, and I hope I've been able to convince you that this population is structured in a much more definite way than we would expect for such a closely related group. The next part, where we ask the question of where this diversity comes from, is a little bit more tricky to answer, and I think it will take many more years and many more people than just me to come to a final conclusion. We do have some ideas, though. The first and most obvious explanation would be that it is based on salinity, since that is what we manipulated at the start of our experiment. Unfortunately, our data does not agree with that. For one, we don't see 3 obvious clusters. There's no way to identify a 20% salt or a 30% salt group. We see strains from all 3 salinities clustering together into heterogeneous groups, like the one that you can see on the slide, where the first number in the name represents the salinity the isolate was grown at. When we check this statistically, we see that the p-value is 0.68, which is not significant, and it means that the population is not clustering according to salinity. When we approach the question from an evolutionary standpoint, there are 2 main processes that can be at play. selection or recombination. By taking a look at the accessory genes, we get the idea that there might be some level of micro-niche adaptation at play. This graph shows the presence and absence of some of the annotated accessory genes. And what we can see are these gene blocks, where specific sets of genes are shared by specific clusters. We also see a couple differences in the clustering of our strains. We have this one group that is composed of several strains from several different clusters, and then those two genomes that I mentioned earlier are also clustering differently in this case. I'm in the process of trying to study these genes specifically a bit more closely, but unfortunately, because we're so limited by the poor annotation, I can really only look at a very, very small part of the accessory genome. Finally, the last option that we can take a look at, or the last hypothesis, is recombination. And the idea is that recombination within the clusters would be higher than between the clusters, and so would maintain monophyletic groups because of this increased gene flow. Unfortunately, this is very difficult to measure with the current tools available. We have some results that suggest maybe recombination could have an influence, such as the reticulation in the gene network, but the programs that are designed to look at recombination are based on a very, very specific hypothesis where they define recombination on the basis of variation. This means that it's typically defined for and designed to run between species rather than within species. And so any conclusions that we draw on the basis of these programs are very, very oppervlakkig. They're very basic, if I can put it that way. They're not as confident as we would like them to be because of how the models fall apart when we're looking at populations as similar as the one that we have. Nevertheless, we're able to conclude that the Salinibacter ruber population in the Saltern has a distinct reproducible genetic structure that is maintained at a global level. We have shown that the metagenome is able to capture the diversity in this population and that the clusters coexist in the environment. We know that this diversity is not due to salinity, and we're taking the first steps towards uncovering the roles selection and recombination play in maintaining and developing this diversity. Thank you very much for listening.
Thank you, Catherine. Well, I must say, for me, always an enjoyable talk, but I am very, very biased at this stage. So let's see if we have specific questions.
I can have one.
Yes, Tuan can go ahead.
Catherine, okay, I go before you.
Yeah.
Okay. Catherine, can you hear me?
Yes, I can.
Okay, all right. Yeah, excellent talk and very interesting work. So yeah, I also think that its recombination within cluster must be more than between cluster, otherwise they won't exist. But what do you think is the selection force in maintaining the cluster other one and also maintaining the overall population? So what is the boundary or defining the boundary of recombination between and within cluster? What kind of force do you think it is?
I mean, that's the question, isn't it? So
yeah, I mean, speculatively, what would we think?
The thing is, it's really tough to say, because looking at specifically the gene functions and looking at specifically what could be causing this diversity, something I've just started looking at. It might be to do with things like environmental sensing and substrate, like decomposition and carbon sources and that sort of a thing. But it is so difficult to prove that definitively as well. Even if if I find a pattern miraculously and I can say for sure that it's this set of genes, it's still very difficult to say definitely that that is the environmental factor that is controlling it, right? So the other thing that with this population that I think is interesting is that I don't think it's one cause in all of the clusters overall. So I think like one of the clusters might be adaptive, and that's why those genomes are grouping together, whereas another cluster might be held together by the recombination. So I think it's a very interesting population. It's a very interesting data set to take a look at.
And Nicola, you've got a question.
Yes, hi, thanks, Catherine, for a really interesting talk and really lovely slides. I love the fact that your bullet points were little phylogenetic trees. I thought that was such a lovely
I felt, I just want to say I'm so glad someone noticed that, because I was so proud of that idea.
Yeah, no, I want to copy it. I want it. Yeah, so I might be betraying a bit of ignorance here about bacterial prokaryotic life history. But I presume that the overall reproduction is asexual, and then you don't know the extent to which there could be recombination going on. And yeah, okay. So as far as I know, the whole astral methodology, one of its assumptions is that there isn't gene flow, as far as I'm aware. Yeah.
Yeah, a lot of the programs do have those caveats, if I can put it that way. But that's why I included the network, for example, that does take that into account, and that
Yes, although that was the other thing, is that for splits tree network, did you need to input a concatenated set of sequences then? Okay, because I think there is a network analysis that uses individual gene trees, and I can't recall the name.
Yeah, I also don't know.
Yeah, but I think it's a sort of like a genomic network analysis that I think maybe it might address that better than splits tree then.
I'll take a look. Thank you.
Yeah, well, I wish I could remember the name to tell you, but thanks for a really, really interesting talk. It looks like a great study.
Thank you.
Yeah, that's one of the things about the prokaryotes, is that you think that they're asexually, but then you have within this population, you've seen the pangenome of the accessory genome. So they have a lot of genes that are only shared amongst a few individuals. And these things are, well, as with recombination, it's a horizontally transferred process. So because it's asexual, they need to get it from one donor. So it's not a shared thing. It is, being able to take it up from whoever is in the community. And there are a number of studies where people are looking at pangenomes, so all the genes together within the species, and having the theory that what the species is doing is they maintain all these genes that they might possibly require over time, and therefore they as a population, they have this access to it, and they can easily start sharing it again. Any other questions? Okay, then thanks again, Catherine, for a very nice talk. And then we will move on to our last talk, which is will be given by Diego Castillo. He's from Tulani Makalañani's group, and he will talk about genome resolved metagenomics reveals widespread microbial chemolithoautotrophic potential in the Southern Ocean. And as some of you might know, but some of the others do not know, this talk is also very topical at the moment, because we have the Tara Oceans ship at the moment in Cape Town that will do some metagenome sampling across the west coast of Africa, and Tulani will be one of the super investigators that will go onto the ship. And so this is a very topical talk also to have in dealing with bacterial genomic diversity. So over to you, Diego.
Thank you so much for the introduction. Can you guys see my screen, the main presentation?
Yes, we can.
Awesome. As Fanas introduced me, my name is Diego, and today I will be speaking about the use of genomics to study the metabolic capacity of marine bacteria and archaea in one of the most interesting environments in the world, well, at least interesting for us, the Southern Ocean. And as Fanas said, this work was supervised by Prof. Tulani Makalañani, and it was actually the last chapter of my PhD. We're still working on it to publish it soon, hopefully. But yeah, I will present what we have so far. Antarctica, the Antarctic Ocean, or also known as the Southern Ocean, are unique places on Earth, and the focus of a lot of research, as evidenced by exciting news that pop up every week almost. I don't know if you guys saw 2 months ago, they found the famous Shackleton ship, which sank 107 years ago at 3 kilometers below sea level, using actually the South African icebreaker, the Archaea 2, which we also used to take samples for the study. The Southern Ocean is so important that actually last year, National Geographic recognized it as the 5th ocean of the planet officially. And just taking a step back, why do we study oceans, first of all? Basically because they approximately contribute to half of global photosynthesis. So half occurs on inland and the other half in the oceans. But the Southern Ocean is a very particular and unique environment. And this is because first of all, it surrounds Antarctica. And obviously all the major ocean basins converge here. It doesn't have any continental barriers. And it's characterized, for example, by the Antarctic circumpolar current, which is the biggest current in the biggest oceanic current in the world. And it is characterized obviously by very low surface temperatures. And this allows actually a lot of carbon dioxide to dissolve and to be uptaken by this ocean. Sorry, I forgot to click. So actually, the Southern Ocean is a major carbon sink, is responsible for the uptake of more or less 40% of human-derived CO2 from the atmosphere. So everything that I've mentioned is about more physical oceanography, and we know a lot about it. But we know a lot less about the biology. At least we know in general that in the world's oceans, phytoplankton and other microorganisms play very important roles. For example, also in carbon sequestration, in various nutrient cycles, and in this way they sustain food webs. And this is of particular interest in the Southern Ocean because it is characterized by many high nutrient low chlorophyll regions. So what does this mean exactly? So for example, on this map that I'm showing you, you can see that nitrate has very high concentrations in the Southern Ocean in the surface. And this is the case with other nutrients such as phosphate, for example. And these nutrients are key ingredients in a way for photosynthesis for phytoplankton. So from this, we would expect a lot of primary productivity of photosynthesis happening in the Southern Ocean. But this is not necessarily the case for many regions. And in any case, there should be much more green, much more chlorophyll, which we use as a proxy for photosynthesis. And why is this the case? So one of the major findings of the Southern Ocean in the past years is that there is no much iron and other trace metals like manganese. And these are very important, obviously, for photosynthesis and other biological reactions. So even though there is a lot of substrate in a way or a lot of food for phytoplankton, there is no trace metals or there is no bioavailable iron. And because of this, there is not much photosynthesis happening, or as much as one would expect. So for our study specifically, I want to highlight, for example, that the lack of primary productivity in the surface will translate into less organic carbon or organic matter in general, sinking into deeper parts of the ocean for other organisms, micro and macroorganisms, to actually use that food. So actually most studies, not just in the Southern Ocean, but in all oceans, are focused on the sunlit layer, which is the part of the ocean where the sun, where light penetrates. And yeah, most studies are focused there because it is very interesting. Obviously, photosynthesis is happening here, but also it's easier to get samples from there. But the dark ocean, or the aphotic zone of the oceans, constitute the largest ecosystem on Earth and still remains largely underexplored. In this ecosystem specifically, obviously, microorganisms also play very important roles, such as carbon cycling and carbon storage. And also microorganisms here help sustain food webs, help feed other bigger organisms and sustain these food chains in these extreme environments. So just going back to this picture I showed you, even when we studied bacteria, for example, not just phytoplankton, the focus has been heterotrophs, that is bacteria that can consume organic matter, right? That is produced by primary productivity, by the phytoplankton. So basically phytoplankton produce organic matter and bacteria eat it. So that's been the simplistic view, let's say. And most of the research is about those heterotrophs. But however, recent studies in some oceanic regions have shown that production of organic matter by chemolithoautotrophic could give alternative nutrient sources, especially in the deep oceans. So just a quick recap for those that are not familiar with this. Chemolithoautotrophic are those microorganisms that use chemical reactions as energy source. They use inorganic chemicals as electron donors, and they use inorganic carbon. So basically, for example, in the oxidation of reduced inorganic nutrients, such as nitrate, so from nitrate to nitrite, that reaction, they will use it to fix CO2, in other words. So as I said, in the Southern Ocean, there is less organic matter, right, for heterotrophs and for other organisms in the deep. And also, as I said, because there is low surface, the temperatures in the ocean surface are low, there is more inorganic carbon sinking to the deep. So there is more inorganic carbon and less inorganic matter, and more organic and less, organic matter. Then that made us think, what about the chemolithoautotrophic in the deep Southern Ocean or in the aphotic zones of the Southern Ocean? And this brings me to the aim and the research questions of this part of my study. The aim was to study adaptations of these microorganisms in aphotic waters in the Southern Ocean. And first we asked what bacterial and archaeal groups are found in this ecosystem and what metabolisms they have evolved to uptake inorganic carbon. So as I said, we used the SA Agalis 2 in winter and spring of 2019 to obtain our samples using a CTD rosette, which is this bottles that you can see in this little video. And that allowed us to take water from different depths. So we took water from 200 meters, 500 meters, 1,000 meters, 1,500, and also the deepest one that was usually 10 meters above the sea level. And then our sampling transect was from close to South Africa all the way to the ice sheet. And this transect went through all the major zones and fronts of the Southern Ocean, like the subtropical front, the subantarctic front, and the polar front. And then after collecting the samples, we filtered the water, as shown in this video. Obviously, using that 0.2 micron filter, we would trap all the bacteria and archaea there. And then we perform shotgun metagenomic sequencing using Illumina. And from there, we did bioinformatics to get all our results. So for those that are not familiar with metagenomics, basically from that one sample, from those filters or from all the DNA that comes from all the microorganisms there, through shotgun metagenomic sequencing, you just sequence everything, like without a PCR, so it's unbiased. And then from those short reads, for example, that you get from Illumina, you assemble them into contexts. And from those contexts, you start grouping them or binning them, as we call, into particular genomes that we call metagenome assembled genomes, or MAGs. So just to show you some stats about our assembly first, in general, we obtained 821 MAGs, or metagenome assembled genomes. And from those, we retained 628 that were medium to high quality. So the assembly was pretty good. And from there, through ANI, we also identified 216 potentially novel genomes. And then when we looked at now genome statistics, we actually saw that, for example, with genome size, GC content, and coding density, these stats were conserved among or within specific bacterial and archaeal groups. So who was there? And we performed a taxonomic and phylogenetic analysis, and we observed great diversity of archaea and bacteria in the aphotic water column of the Southern Ocean, as we call it. So this outer ring represents the different depths by the shading of the blue color. And we obtained 114 Archaeal mags, mostly from Poseidonia and Anthrososphaeria. These are common, well-known, the ammonia oxidizing Archaea. They're called also Archaeota or Archaeota. And then we obtained lots of bacterial mags belonging to 18 different classes. And we saw a lot of diversity of well-known gamma protobacteria, for example, but also we saw less studied groups like SAR324 or Acidomicrobia. The interesting thing was that when we performed the functional annotation, we observed, and it's indicated by this last outer ring with these little color bubbles, that 167 mags had genes for inorganic carbon fixation pathways. So I chose some representative ones to show you guys. Most of these microorganisms seems that can fix inorganic carbon, and they rely on the Calvin cycle and the reverse TCA, as shown here. But also interestingly, we saw that they also had certain genes to oxidize those reduced inorganic nutrients. So as you remember from the beginning, when you have those two things, the carbon fixation plus the capacity to oxidize those inorganic nutrients, that means that it could be chemolithotrophs. So we found genes for example, thiosulfate oxidation with a SOX complex indicated in red, and also I highlighted the red mags, ammonia oxidation with the AMO-A gene, and nitrite oxidation, for example, oxidation. So we also found, and this was consistent with current literature, certain chemolithotrophs that have previously found in the dark ocean, like SAR324 and the Archaeota or Nitrososphaeria at this level. And interestingly, we also saw that certain genomes or certain mags had the capacity or the genes to metabolize carbohydrates, as shown here by the carbohydrate active enzyme genes. And some of them were also chemolithotrophs. So that means they have a very versatile metabolism. They could be mixotrophs, so they could fix their own carbon, and they can also produce, and they could also eat or use organic matter. As I told you, the interesting thing was that 25% of these mags have the capacity for chemolithotrophy, which is insane. So we actually tried to map them and see where they came from. So originally we had 44 samples, 44 metagenomes from the stations I showed you from Antarctica all the way to South Africa. And then again, the blue corresponds to different depths of the aphotic water column. And we mapped those reads back to the original metagenomes, the original samples, and we saw that many of these chemolithotrophs were highly abundant everywhere throughout the aphotic water column and also across our huge, almost 3,000 kilometer transect. So this means that chemolithotrophy might be spread everywhere in the dark Southern Ocean. And this corresponded to our hypothesis that maybe because there is more inorganic carbon sinking into the deep and less organic matter, these microorganisms have evolved chemolithotrophic everywhere in the Southern Ocean. Finally, we just wanted to understand better the mechanisms behind this. So we performed a metagenomic analysis, and I won't bore you with the details, but I have just zoomed into these genes that help chemolithotrophy, as I mentioned, for AMO-A for ammonia oxidation, or the SOX complex for thiosulfate oxidation. So we actually observed that clusters of variable orthologs proteins from diverse phylogenetically distinct mags were identified. These included, for example, for the SOX complex, different bacteria from different phyla even, and for the AMO-A, for the ammonia oxidation, we even saw different, we saw bacteria as well as Archaea. So this suggests that the maybe we hypothesize, and we have to do more analysis to corroborate this, that the ability to oxidize inorganic nutrients in the Southern Ocean has been shared via horizontal gene transfer between bacteria and archaea, and also within bacterial phyla. So just in summary, we reconstructed more than 200 potentially novel genomes, and we identified that bacteria and archaea harbored an array of metabolic strategies to fix inorganic carbon through chemolithotrophy. And we saw that this metabolic strategy might be widespread everywhere along the Southern Ocean fronts and zones, and also throughout the aphotic water column. And these genes that allow chemolithotrophy might be horizontal laterally transferred due to the pressures of these unique environments. And I would just like to acknowledge my supervisor, Oliver and Tiffany, that participated in this project. Everyone in the research group, the day they took this picture wasn't here, but this is everyone else, and all their funding bodies and the university. Thank you very much.
Thank you, Diego, for a very interesting talk and telling us something about an ocean at our doorstep that nobody knows much about. So again, we're open for questions. Anybody that would like to ask a question, welcome to raise your hand and just unmute and ask the question. Lonnie?
Thank you, Farnes. Diego, thank you very much for your presentation. It's pretty amazing to see. So I'm going to talk about the supervisor, how far Tulani has come, who now has students doing all this work. And yeah, it's you guys are doing really well, and we're very proud of Tulani as a former IMBMer. All right, so coming back to your work, if we look at that phylogenetic tree that you had of all the bacterial species that you identified, one question I had was, what program did you use for binning? Because it seems like you have very many very closely related species in the tree.
Yeah, let me just put it up again maybe quickly. Okay. So for binning, we used a combination of Metabat, Concoct, and escapes my mind now. Well, we used 3 different binning algorithms, and then we used that tool to dereplicate. Then after obtaining all of these ones, first 800, now 600, we did dereplicate further, and we obtained much less than we can see here. And I see your point, probably you saying these ones are too similar, maybe it's one genome, right? But the idea of this tree was to observe the distribution also of maybe similar genomes throughout the aphotic water column. So we have another part in the paper that we just work with the dereplicated genomes, but here we wanted to show that even genomes that maybe are 95% similar, the same species, for example, they come from different parts of the aphotic water column.
Okay.
Any additional questions that you would like to ask?
I've got one out of ignorance, and it's amazing diversity, but is this unique to the Southern Oceans? Would we expect a similar pattern if we looked at the oceans across the world, if we looked at deep terrestrial systems? I'm thinking about the alkaline vents and pools, which are similar to the ocean. Would their biodiversity be different? So
one of the studies that I showed the screenshot in the beginning in Nature Comms, I think it was, they showed last year. they did a similar study, but across different samples from different oceans. I think it was the Pacific, Atlantic, and yeah, I think it was the Pacific and Atlantic. And then they already observed that there was chemolithotrophy in certain mags that they had. But what was interesting for our case is just the sheer number, like 25% of the mags that we recovered had this ability. And this could be because of the uniqueness of the Southern Ocean, where there is more inorganic carbon, more CO2 taken into the deeper parts of the ocean and less organic matter sinking. So it has been shown in other oceans, but the interesting part about this study is that the amount or the abundance and the widespread of this metabolic strategy.
Thank you. Anybody else that would like to pose a question? Well, it doesn't seem like that. So, Diego, thank you very much also for your contribution. And it just sort of, in my mind, created this idea that, yes, we need to give these mags and things formal names so that we can actually start talking about it in the literature and not reinventing the wheel. So I think, I'm deeply involved in the SETOT initiative, but I feel, this is the right thing to do. And my feeling is that once you have a name, you can have a community that works on the description of this organism. So it's not like you need to describe everything in detail up front. Once you have a name and you have good diagnosis, you can actually move on and then expand the literature. And I think everybody would benefit from that. So also thank you to you for the work that you've shared with us. So thank you for everybody that's joined. Usually at the end of these meetings, we have a time where people can ask questions of general interest. So let's see if there is anybody else that would like to raise an issue that is sort of importance or interest. Nicola says, thanks for all the speakers for a fantastic session. I was glued to my screen the whole session. Oh, that shows you what bacteria can do. I'm very biased. So, yes, I think one of the things also that we know working with bacteria is that, you have a very quick reproduction time. Evolution is in a fast pace and things, and you can actually get a whole genome and, work with whole genome.
Did
so,
you get to study recombination?
Yeah. So thank you very much for that. And for the ISME members that have joined us, thank you also for joining us. And if you're interested in the kind of things that we are doing at the Society for Systematic Biology, please add your name to our the website where you can get reminders of the monthly meetings. And as I say, there won't be a bacteria next month, but in the future, we will definitely make sure that we have another one to work, to get some idea of the prescriptive linguistics and not the descriptive linguistics. I quite like that, that comparison. So if there's nothing else, let me just see, it's the same comment. I think that's the end of our session today, and thank you very much to all the speakers and to all the people that have joined us. For those that have missed it, we were good this time. We've recorded the session, and it will soon be posted on the SASSB's YouTube channel. So you can have a look there, and you can also refer other people to go and have a look.
There's a question on the chat stop to see.
How do you describe to the Catherine? Okay, yeah. There's the link. It's if you go to the FABI website, you will see the link to the SASSB and that's where you just add your name, and then we will, on a monthly basis, send you the invites for the new, sorry, for the new webinar. And the typical date for the webinar is the first Thursday of the month. So with that, thank you very much for everybody that joined, and have a lovely evening. I hope that you have light in your lives.
Thanks everyone. Thanks Farnes and the team as well for organizing. Bye.
It's a pleasure.
Thank you.
Bye.
Bye everyone.